so much, and thank you all for joining tonight, and thanks for the invitation. I'm really excited to talk to you about my work at Fred Hutch. I'm a postdoc in Toshi Tsukiyama's lab, and I've been trying to understand how cells wake up from like this dormant, sleeping type of cellular state, which I will tell you all about momentarily. Oops, hold on. There we go. Sorry about that. For some reason, my arrows were being weird. Okay, so what is a quiescent cell? So actually, surprisingly, most cells on Earth, whether it's like bacteria or like archaea or something, they actually spend most of their lives in quiescent state. So they're not actively dividing. And so we think of life usually as like, cycling through the cell cycle where we have like this nice growth stage and then DNA replication and cell division. But in many cases, cells can actually exit the cell cycle into this state called G0. And there's a ton of different types of G0 states. Some cells enter G0 from different branches of the cell cycle, and there are two different major types where you have a reversible G0 and then an irreversible G0. And for this case, these become generally senescent, or they terminally differentiate, so they no longer divide. But quiescent cells are special in that they can actually enter quiescence and then exit quiescence again and go back into the cell cycle, so they can actually go back into cell division. So that makes them really special. And so this is going to be the focus of my talk today. Arrows are being weird. Okay. But one of the key practical uses of quiescence is that it can help cells survive long-term harsh conditions. So this can be like microbes, or for long-term survival, we have like hematopoietic stem cells that can be there for years while we're in an organism. But in terms of why some of these cells become quiescent, they could be starved, they could be desiccated, like probably heard of tardigrades, where the whole organism becomes dormant. That's a bit different from quiescence, but a lot of similar principles happen. And then we get like contact inhibition, where a lot of cells will grow together and then need to stop dividing at some point. But one of the health-related reasons why we're interested in quiescence is it's important for maintaining the stem cell niche. So you have a population of stem cells that need to either self-renew or differentiate, and they need to enter and exit quiescence in order to do that. And so I'm actually interested in both of these types of quiescent strategies, so surviving harsh conditions as well as stem cell niche maintenance. But I'm going to be focusing today on this top part where actually starvation is driving quiescence. And to answer this question, I actually am using the model organism budding yeast. And yeast can actually enter quiescence. So this is the same yeast that we use to like bake bread, brew beer. It's a fantastic model organism for answering this question, because what we can do is we can grow the yeast in rich medium, which I'll refer to as YPD, and actually grow them for 7 days. And at the end of the 7 days, we'll get this mixture population of either dead cells or non-quiescent senescent cells. And then we get these nice quiescent cells that actually store a lot of different sugars that make them quite dense. And so we're able to put them over a Perk-Hall gradient and actually isolate the quiescent cells at the bottom of the gradient. And this is really nice because it's actually really it can be very hard to study quiescent cells, like from the stem cell niche, for example. It's hard to keep them in quiescence and like study large quantities of them. Single cell sequencing has really been helpful for answering a lot of those questions. But the nice thing about this is where I'm actually able to harvest tons of quiescent cells and actually purify enzyme complexes from quiescent yeast cells and actually study them. So that's really been helpful for understanding molecular mechanisms for quiescence. But one of the emerging hallmarks for quiescent cells in both yeast and mammals is that these cells create a repressive chromatin environment. And this is actually I'm going to take a moment to kind of go through chromatin real quick, just to make sure that we're all kind of up to speed and on the same page. So just to kind of take a step back, DNA is packaged into chromatin. You'll see like these nice, the DNA is wrapped around these histone proteins at about 147 base pairs each, and then they kind of get further packaged and can form chromosomes eventually. And I'm going to be actually focusing a lot on chromatin and these nucleosome positions. And to kind of zoom in a little bit more, we're going to look at the basic unit of chromatin here, which is the nucleosome. And this is a beautiful structure from Carolyn Luger's lab. And you can see that we have 4 histone proteins, and they get about 147 base pairs wrapped around them. And these also get decorated with histone modifications. So like chemical groups will be tagged on these histones. And that's going to be really important for regulating gene expression. And I'm going to be talking about one important histone modification that will occur on H4 tails, which are not in the structure, but it's acetylation. So these acetyl groups get added to the H4 tails. And I'll be mentioning that later. And I will also be mentioning how these nucleosomes get moved around. So actually, these are really important for regulating gene expression. And there's a whole class of proteins called chromatin remodeling enzymes, where they use ATP to basically move, assemble, and eject nucleosomes to help promote or regulate any DNA transaction like DNA replication, gene expression, etc. And so there are a lot of different types of chromatin remodeling enzymes, and some of them, like I said, they can assemble nucleosomes. They will actually move nucleosomes around, so they'll create more open chromatin, for example. And I'm going to be talking a lot about switch-SNF today. And then there's also nucleosome editing, where they can actually add different histone variants, for example. And so there's a lot going on in these nucleosomes, and this is actually very important for quiescence. And chromatin actually changes so much during quiescence entry. So a fantastic paper from a former postdoc in our lab, Jeff McKnight, he pretty much set the groundwork for understanding how chromatin changes during quiescence entry. And so he found that quiescent chromatin is very repressive. So transcription factors, so this is just a little picture of a gene, and I have these little nucleosomes in these little dark circles, and then they're blocking transcription factors in RNA polymerase from being able to bind to the promoter, which would normally be here. And what he found was actually, so I mentioned earlier, histones can get acetylated, and that's important for open chromatin. The acetyl groups get removed during quiescence. And so that makes chromatin even more repressive. And then these promoter regions, which are usually depleted of nucleosomes, so they are nucleosome depleted regions, or NDRs, they get blocked by nucleosomes. And they have higher levels of histones bound to the chromatin. So you have more nucleosomes there. And then as a result of all of this, we get little to no transcription. And so Jeff found a key regulator for promoting this repressive chromatin environment. And he found that there was very little, pretty much no transcription in quiescence. What I'm interested in is, how do cells turn on the genes? How do they wake up from quiescence? And so, here's my question: how do cells exit quiescence? And so one of the interesting things is that I found very soon after I joined Toshi's lab, I found that quiescent chromatin, or quiescent quiescent cells can actually activate their genomes really quickly. So to look at this, I basically was able to purify quiescent cells after that 7-day time course I showed you earlier, and I just added them to fresh media, and at 5 and 10 minutes, and then I can even take them out to 240. This is when they go back to regular cell cycle. What you can see here is when we look at nascent RNA-seq analysis. So basically, I'm labeling the RNAs with a base analog and IPing the new RNA that's created. And you can see in quiescence, there's very little transcription happening. And within 5 minutes and 10 minutes, you can see tons of transcription happening. And I forgot to mention earlier that these yeast quiescent cells can stay in quiescence for a really long time, like even up to a year. And I even personally did a 6-month exit time course. And so it's pretty remarkable that they can actually exit really quickly. This is just a 7-day culture. It's just something to keep in mind. So this kind of, this basically made me wonder was the transcription machinery already bound to the genome during quiescence? And so I wanted to answer this question by doing RNA polymerase ChIP-seq analysis. So basically RNA polymerase, which is responsible for transcribing mRNAs, I'm able to basically immunoprecipitate this from chromatin and then sequence the DNA that's bound to the RNA polymerase. And so here we're looking at heat maps. So the dark purple has high signal. So this means that there's a lot of RNA polymerase binding. And then the orange means there's less. And I've just done like little k-means clustering to kind of group these and look at the different patterns of RNA polymerase binding. And so what I hope you can appreciate that is in quiescent cells, there's actually very little RNA polymerase already bound. I mean, maybe a little bit in some cases, and there is still some transcription. But what I hope you can see is that there's still like a huge change, very similarly to the RNA-seq analysis. But what this tells me is that RNA polymerase, even though transcription turns on so quickly, it has to really pretty much de novo load onto chromatin. So it's not necessarily poised, for example, in quiescence to allow for this really rapid transcription activation. And these lines here are just these meta plots for the different time points. And what I hope you can see is, again, there's like tons more transcription that happens during quiescence exit. And also this is just normal cycling is log cells. And it's kind of remarkable that even more genes are turned on in the very early exit time points compared to log. And I think what may be happening is that cells have to make a lot of transcripts during exit. I mean, during entry, you have to get rid, so a lot of proteins are degraded. So you have to like remake a lot of things during exit. So it's pretty important to have this early spike in transcription here. And what this is called is hypertranscription, when you go from a global off state to a pretty genome-wide on state. And hypertranscription is pretty conserved throughout a lot of different processes. So it can occur during zygotic genome activation or lymphocyte activation. It can happen in a lot of different cases. And it's really fascinating that you're going from this like very low level of transcription to a high level of transcription. So how is this regulated? And what are some of the key regulators for this process? And so this is kind of where I'm going to lead into is, I found one of the key regulators, which I recently published, and I'm going to kind of go through this. One thing I wanted to look at when I first started trying to figure out how to answer this question was, what does the chromatin look like between quiescence and that early exit time point, the 10-minute time point? And so I used this protocol or this experiment technique called MNase-seq. So MNase is an enzyme that will digest DNA that's not protected by nucleosomes. And so you can actually get a really nice readout of how the nucleosomes are positioned in the genome. The other really nice thing is we can use 2 levels of MNase digestion to kind of get how stable is the nucleosome as well. So we can actually use a lower level of MNase to then look at nucleosomes that are like considered more fragile or labile, because when you see them, you can see this nice peak here. And this is actually the whole yeast genome. So this is an average across 6,000 genes. So this is a pretty significant peak here. And it actually is gone when we add a higher level of MNase. So this suggests that this is actually a not very stable nucleosome that could. And so what was really interesting to us is that in quiescence, we have this nice stable nucleosome peak here, and then at 10 minutes, we see it go away. And so this suggested that there was something being moved around, like a chromatin remodeler, as I mentioned earlier. And one of the major chromatin remodeling enzymes is called RISC, which is a big protein complex. It stands for remodeled structure of chromatin, and it's conserved from yeast to humans. And its major job is to basically create these nucleosome depleted regions. It's going to slide nucleosomes out of the way from the promoters and then basically give way for these transcription factors to interact with the DNA. And this is also a member of the switch-SNF class of enzymes, and it's known in humans as PBAF. And importantly, these switch-SNF classes of enzymes are mutated in 20% of all cancers. And so you can see here in this nice review article, which is from Sigal Kadak, she basically mapped out where all of the different chromatin remodeling subunits are mutated in all these cancers. And the complex that's most similar to the yeast RISC complex is this PBAF complex. And so I found in Quiescence that RISC is actually bound in a very unique way. It's bound to the majority of NDRs in quiescent cells. So you can see here in this ChIP-seq analysis where you have this nice stripe of RISC binding all the way down through almost nearly all Pol II genes in the yeast genome. And so we created a strain where we can completely obliterate RISC by using an auxin degron system, so we can rapidly degrade the complex. And so when I do this, I'm just going to quickly summarize this because a lot of what RISC is doing is known in log cells or cycling cells, but we had no idea what it was doing in Quiescence. So what it does in Quiescence is it actually causes the nucleosome depleted regions to narrow. So like the nucleosome positions are for getting closer to the promoters. The positioning in gene bodies actually gets worse. And then also the histone occupancy is increased. So when I do a H3 chip, for example, I see more of the histone at the promoters in Quiescence. And when I get rid of RISC, it actually really harms transcription activation. And so normally when we have transcription activation during Quiescence exit in the presence of RISC, we see this nice increase in RNA polymerase binding. This is a Pol II ChIP-seq. And you can see again, like at 30 minutes, it goes back down. See, this is that hypertranscription event that happens. However, in the absence of RISC, we have less transcription even in Quiescence. And then as in these early Quiescence exit time points, the polymerase cannot load and transcribe at 10 minutes. And then even at 30 minutes, it never reaches the wild type condition. So what this is suggesting is that RNA polymerase is required for RISC. RNA polymerase requires RISC to actually initiate and transcribe during Quiescence exit. And this is actually just looking at initiation, but I noticed some interesting profiles at some subsets of genes when I was looking at Quiescence exit and RISC's role there. And there are actually a lot of different ways RISC ruins transcription, and I'm going to kind of go through those here. And so, so here's the first part where RISC is blocking RNA polymerase from initiating in Quiescence exit. And this is actually pretty well characterized for RISC. So the next few are actually going to be more novel roles for RISC activity and transcription. So what was really interesting is I mentioned earlier that RISC binds to chromatin in a more unique way during Quiescence compared to logs. So in log cells, I'm not showing it here, but basically log RISC mostly binds to the nucleosomes on either side of the promoter. It's actually not normally in the nucleosome depleted region unless it's a really wide NDR. So it was really cool when we saw that RISC was binding to NDRs because it kind of suggested that RISC might be kind of sitting there inactive. And so I looked at what happens to RISC during exit, and you can see it's actually kind of moving out towards the gene bodies or away from the nucleosome depleted region. And I personally think that that means that it's getting activated. So I think it's sitting in Quiescence inactive, and it needs some kind of signal to activate during exit. And so I thought maybe that signal could be RNA polymerase itself. So at this is a subset of genes, like I mentioned earlier, there's a bunch of different classes. And so I was able to actually block transcription, which blocked RNA polymerase from loading. And that actually caused RISC to get stuck in the nucleosome depleted region. So you just use this drug in the black line to block transcription, and you can see a lot of RISC is gone, but a lot of it is actually stuck in this NDR. So that actually suggests that maybe Pol II can actually help RISC get activated, and then RISC can help polymerase transcribe. And so to look at that, I looked at some of these genes, and I looked at RNA polymerase localization in the absence and presence of RISC. And so here's where RISC itself is binding. So you can see there's like this nice peak here. And then when I get rid of RISC, polymerase kind of gets stuck at this transcription start site. You can see the peak is right there. And then in the presence of RISC, you can see it's like kind of elongating across the gene body. And you can see it's kind of, RISC might actually be helping to activate polymerase elongation, which was actually pretty new though there have been really nice studies from the Govind lab that suggest that RISC is involved in elongation. And this was really nice to see because we're looking at a quiescence exit where we're having no gene expression and then lots of gene expression. And so basically what happens is RISC is preventing RNA polymerase from being mislocalized. So it actually helps it, not only helps it load, but it helps it go to the right place. And another class of transcription or another class of genes that I noticed was that it was really weird when I looked at RNA transcription or like RNA-seq analysis, as I showed earlier in the talk. There's actually, so I take the log2 ratio plus and minus RISC, and so the yellow is actually showing what is increased in the RISC depletion condition. And what I hope you can see, and oh, I forgot to mention, this is also like across the genes. So this transcription start site is right here. So up here is like before transcription normally begins. In the absence of RISC, we're getting upstream transcription start site activation. So you can see a lot of yellow here, and it doesn't really elongate very well. So that actually suggests that RISC is also directing RNA polymerase to start in the right location too. And here's just one example. So I'm going to walk through this. It looks a little complicated, but I'm going to walk through it and we'll be able to look at this. So these little peaks here are nucleosomes. And then this is a gene called PTP3. It's transcribed in this direction where the little arrows are pointing. And the pink is just showing this is a RISC depletion condition. And we're looking at nucleosomes, which are the MNase. We're looking at RNA-seq, which is labeled by 4TU. It's the type of nascent RNA-seq analysis. And we're looking in quiescence and in 10 minutes. And so let's start here at the RNA level. So what I hope you can see is that in the RISC depletion condition during quiescence exit, you're seeing an overall lower level of this transcript. But what I hope you can also see is that this transcript is actually starting before the normal condition. So this is the normal condition. You're getting really nice sharp peak here, and then you're not really getting anything there. And so this was suggesting, this is basically reflecting what we're seeing here in the heat map. And then when we look at the nucleosomes for this, so in the absence of RISC in this 10-minute time point, you can see that this nucleosome depleted region is actually filled with the nucleosome, unlike in the wild type condition where there's this nice wide area for RNA polymerase to properly load. And so RNA polymerase would normally load here, transcribe this. But unfortunately for these cells, RNA polymerase cannot load here, so it's going to go to the next spot and create an NDR. I actually don't know why this NDR is lower, and which is really interesting. And so you can actually see RNA polymerase potentially loads and transcribes there. So that was a lot of work, or a lot of things to kind of say that, you know, basically RISC is kind of controlling this chromatin environment to not only help increase transcription, but also basically create accurate transcription. And I just have this here, the RISC here showing that this is a RISC target. So it's directly showing, it's directly functioning at this gene. The last part of the RISC story is that RISC depletion also causes a an increase in antisense transcription. So basically, antisense transcription is when transcription happens in the wrong direction. So normally you'll have like an example gene where you have like transcription going in this direction. However, if your nucleosome depleted region is too small or absent, you might not get transcription at all. And in this case, so here in the absence of risk, we're looking at just two different clusters of genes that I just kind of made. And I saw like a lot of antisense transcription lighting up here. So these are all strand-specific RNA-seq data sets. And so you can see that there's a lot of antisense transcription here. And then in some of these genes in this cluster, there's actually reduced sense transcription. So in this case, I think this is a little confusing. I just realized the purple is actually low in this heat map and the orange and yellow are higher. I apologize for that. So what I hope you can see is that in the antisense, we actually have a lot more transcription in the absence of risk. And then in the sense we have kind of a mixture. It didn't fall 1-to-1. And then there was this other class of genes where I saw actually reduction in antisense transcription. And then sense was kind of the same. It didn't fall perfectly correlative. But what I hope you can see also is that when we have the genes with a lot of antisense transcription, those nucleosome depleted regions in the sense direction are actually much smaller. So that's what's happening here in this little cartoon. So for this guy up over here, we're seeing these nucleosome depleted regions getting kind of squished. So that's what this pink line is in the absence of risk. And then we're also getting an increase in antisense transcription here. But when we don't have antisense transcription increased, we have more or less similarly spaced and nucleosomes. So that suggests also that risk is preventing aberrant antisense transcription during exit. And then to kind of summarize this risk part of the story, we have the idea basically that risk is protecting the transcriptome from bad gene expression. So in quiescence, we have risk sitting in the NDRs. And I showed you earlier that risk kind of moves out of the NDRs and moves these nucleosomes out of the way so that RNA polymerase can load, but it also like spaces the nucleosomes properly so that RNA polymerase can plow through the gene properly. And then when we don't have risk, we have lack of transcription starting. We have RNA polymerase not getting through to the gene body very easily, or it starts too early. So we had that upstream transcript that started. And then we also have an increase in antisense transcription in a lot of genes. So without risks, the cells are very sad, and they actually exit quiescence quite slower. I didn't put it in the talk today, but they have a quiescence exit defect as well. And so, I showed you that like transcription activates really quickly during quiescence exit, and that risk is a major regulator for promoting gene expression during exit. So this was all published last year in a paper in eLife. But, I figured there kind of had to be other factors that were required for regulating chromatin during quiescence exit. So I wanted to look at other chromatin remodeling enzymes. So I'm just going to briefly go through unpublished data. This is all very new. And I have a lot of questions, so I don't have a lot of answers, but I think it's pretty interesting. So there's this other chromatin remodeler called Fun30, which actually literally stands for Function Unknown Now 30. And we do know the function of Fun30. It's actually involved in DNA repair, and I'll go through that in a minute. But it's kind of a fun name, silly name, but it's called Fun30. And it binds to nucleosome depleted regions as well. And I've accidentally left this here. This is actually just risk, again, just showing it at the nucleosome depleted regions, which are upstream of the transcription start site. And so we had done, actually a wonderful grad student in the lab had done a beautiful chromatin remodeler screen where he chipped every single chromatin remodeling enzyme known in yeast in quiescence. And Fun30 actually bound very similarly to risk. And so this really was curious to us, like, why would both remodelers be at the same locations in quiescence? So I'm kind of trying to figure out what Fun30 is doing. But to kind of, before I go through and before I go into that more, I wanted to kind of give a little overview of Fun30. So it's actually a pretty understudied chromatin remodeling enzyme. So RISC and switch SNF are super popular. I mean, there's a whole company devoted to, probably more devoted to, like looking at therapeutic targets and chromatin remodeling, like switch SNF complexes. But Fun30, we don't know as much about it, but we do know that it's conserved in mice and humans, as well as yeast. It's upregulated in pancreatic and breast cancers. And as I mentioned just a second ago, it's involved in DNA repair and actually maintains heterochromatin, which is silenced chromatin. And it's a pretty special and nucleosome remodeler. It can actually create nucleosomes de novo. And there's a beautiful cryo-EM structure from Carolyn Luger's lab that just came out last year of the human form, which is SMARCAD1. And so I kind of wanted to figure out, what is Fun30 doing during quiescence and quiescence exit? First, I wanted to see how it binds compared to log cells, so cycling cells. And again, it's very different between quiescence and log. It's actually and like I said earlier, it's in the NDRs. Normally, it's not really bound to a lot of transcription start sites in log. So it's kind of forming a special binding pattern in quiescence. So it might actually be having a new role in quiescence. And what was interesting is, so here I actually deleted Fun30 in quiescent yeast, and I wanted to see how quickly do they exit quiescence. And so what we're looking at here is flow cytometry, looking at DNA content in the cells coming out of quiescence. So this is about 2 hours and then 3 hours after quiescence. And so in wild type cells, you can see at about 3 hours, we have them stuck in G1. And so we're looking at 1N here. And I was actually just going to look at G1 for chromatin purposes. But I was like, when I did this flow cytometry, just to make sure that they were actually in G1, I saw that Fun30 like exited way faster. Like you can see that. So this is here showing is that they're actually like in S phase. And then some of them are already in G2M at 180. So this was quite remarkable, because I have not seen a mutant that actually exits faster from quiescence. Like, why would cells need to exit, or why would this actually suggest that Fun30 normally represses quiescence exit? Why would you want to do that? I still don't know. If you have ideas, let me know. But this was really cool and interesting. Yeah, so basically this suggests that Fun30's job is to actually slow down quiescence exit. And so I wanted to look, so, Fun30 is a chromatin remodeler. What's happening to the chromatin? So I wanted to look at just total histone H3 occupancy, meaning I just chipped H3 in quiescent cells in wild type and in the Fun30 deletion strain. And what I hope you can see is actually there's less histone H3 in quiescent chromatin. So these chromatin is basically becoming more open. And so I think this suggests that actually Fun30 is stuffing those promoters with nucleosomes normally. And then I wanted to look, so I mentioned acetylation at the beginning of the talk, and I was curious about whether his Fun30 would be increasing acetylation because it was creating open chromatin, and maybe there's another role. I will actually tell you more why I looked at that. But basically what happens is when I chip H4 acetylation in quiescence and presence and absence of Fun30, you can actually see there's an increase in total histone H4 acetylation. And this was really interesting because that again points to Fun30's role in actually making repressive chromatin, which is actually very different from what it normally does in log, because in log its role is to repair DNA. It doesn't bind genome-wide, so it's actually taking on a new role during quiescence entry. And again, earlier I mentioned Jeff McKnight and his fantastic paper. This is just the graphical abstract from his paper. And so he found that during quiescence entry, again, as I mentioned, acetylation is removed and transcription is turned off. And this is all mediated by this histone de-acetylase, or HDAC, RPD3. And what I thought was kind of cool was when I looked at his RPD3 chip-seq data set, it actually overlapped pretty well with Fun30, like it's going to the NDRs again. And so, and between this and the fact that H4 acetylation increased during quiescence or in quiescence in the absence of Fun30, I'm thinking, now I don't have the answer to this yet, but I think maybe Fun30 could be recruiting RPD3 as well. And there's a really nice paper that came out a few years ago that showed, so here we're looking in mouse yeast cell chromatin immunoprecipitation. And so they IP'd SMARCAD1 and they actually see HDAC1 interacting here, suggesting that these two guys are interacting. So maybe in quiescent yeast, these interact as well. And so why would cells need to slow down exit? So this is very preliminary data. And I'm thinking perhaps, so when you exit in quiescence, you can't actually repair your DNA. There's no DNA replication. It's actually shown that DNA repair, we know when it starts again during exit. So it's possible that Fun30 might need to slow cells down to exit in order to help cells repair their DNA before they can divide. So here's a very, very preliminary, not perfect serial dilution assay, but I'm still pretty encouraged and excited by it. So yeast, we can basically spot onto plates and have a nice dilution, tenfold dilution across the plate. And so we have YPD, which is our control plate. And then I can add this DNA damaging agent, fleomycin. And you can see these basically are just further diluted yeast. And then I have this nice control, which is involved in DNA repair. And you can see it doesn't grow on the DNA DNA damaging drug plate. And I can actually compare Q versus log cells, and cells coming out of quiescence in the Fun30 mutant are actually sicker than in wild type, but also sicker than in log. So there's like fewer little yeast spots in the coming out of quiescence than they are coming out of logs. So this is sort of suggesting that maybe Fun30 quiescent cells are sensitized to DNA damage, and maybe that's why the cells need to slow down quiescence exit. But there's a lot of open questions, and I'm really excited to hear some ideas maybe from the audience if you have any suggestions, because this is definitely ongoing work in progress. But I think, I do have one more slide. Sorry about that. So there are some open questions. So as I mentioned earlier, that fragile nucleosome, both risk and for Fun30 are bound here. So maybe they're playing some kind of opposing role at this location. Maybe Fun30 is placing this fragile nucleosome and risk is removing it. And then I still don't know what happens yet to transcription activation. And then again, maybe Fun30 is actually helping to recruit or activate RPD3. And with that, I would like to thank everyone in the Tsukiyama lab, especially I have a high school student, Jonathan Mulenga, who's fantastic. And then I have a few past lab members who have contributed, Sam's, especially for the Fun30 part. And then I also had some nice funding during my postdoc. And then I mentioned at the beginning that we're interested in stem cells and quiescent stem cells. And I have a whole other project on mouse quiescent neural stem cells that I'm just starting. And these wonderful people in Patrick Pattison's lab have been helping me with that. And if you want to get in touch, here's my info. And thank you all for your attention. And I'm happy to take any questions.
That was fantastic. That is super, super cool stuff.
Thanks.
All right, so we will just jump straight into the questions. I'm going to bet that you can tell which ones I asked versus which ones were asked by one of our audience members who apparently has done some work related to this. So we'll jump straight in. So could a better understanding of quiescence facilitate something like the fantasy version of long-distance space travel, so like people being kept in stasis, that kind of stuff?
Oh my gosh, that's such a cool question. I hope so. I have no idea. Okay. I mean, I hope so. That would be so cool. I mean, one thing I want to do is, I mentioned earlier that I had like 6-month-old yeast cells. I'm really looking forward to like seeing what's happening to the chromatin there. And, maybe we can learn something about like suspended animation, or I'm sure the chromatin has to be repressed during that stage too, because you want to turn off transcription so they don't use up all the energy. So,
Yeah, exactly. Okay,
yeah,
cool.
such a cool question.
Are you looking at persister cells in infectious bugs?
That would be cool. You know, Candida, which is an infectious yeast, those cells I've learned actually can become quiescent and actually are resistant to therapeutics. So it would be really interesting to see if we could maybe target certain maybe chromatin remodelers to block them from exiting, to like either keep them in quiescence forever, or maybe use Fun30 delete to drive them out of quiescence, and then maybe those therapeutics would work on them if they're
Yeah,
leaving quiescence. So that would be interesting.
yeah, that would be interesting. Do quiescent cells require some kind of food to survive? So, like, basically like if a cell is quiescent, how does it survive? That's
That's a really good question. So for yeast, they can just sit in water without food. It's really crazy. They basically are able to just, stay long term without any food. Basically they eat upon certain signals, like when they have nutrients and it's safe to start eating again.
okay. All right. Let's
Yeah.
see. Did you use a phosphorylated RNA pol antibody for doing your IP?
Oh, that's a great question. Not in this. I have done that and to see if it's activated. I actually don't think I did it for the IP. I've done it for Western. I can't remember the result if I did it for ChIP-seq analysis to see if it was to see where I think they're asking like serine 2 versus 5 phosphorylation, probably to see if there's initiated and elongated polymerase. I don't think I did genome wide. Um, that's a good question.
Okay. So you talked about hypertranscription. Are there any known issues with things like increased rates of this is my question, so I don't know the right terminology, but like bad transcription or something like that? Like, I guess is there a downside to the hypertranscription?
Yeah, absolutely. And it's interesting, like if one area that I'm really interested in is and I'm wondering if Fun30 transcription will be affecting this, is like if we don't shut down this huge amount of transcription before DNA replication starts, you can get DNA damage happening, like R loops can form, and like the cells might not be able to repair their DNA. And so it can be actually kind of dangerous, especially that first round of DNA replication, because
Mm-hmm.
they have to repair their DNA for the first time. And if you're having a lot of transcription during that process, it can be pretty scary. It's pretty
Yeah.
dangerous for the cells. So that's one thing that I think can be bad. Or if you get a bunch of like bad transcripts, you can probably get like non-stop proteins, for example, like
Mm-hmm.
if you get run-on transcripts, or transcripts that start at the wrong place, you might not get right proteins created. I'm not
Yeah.
sure, but it'll be really interesting to look at all of that for sure.
Okay. Very cool. Do you also see an increased binding of HP1 and associated heterochromatin proteins absence of risk complex?
I like that question a lot. I have not looked at that. Yeast don't have HP1, but as I mentioned earlier, I'm moving to this mouse neural stem cell model, and so
Mm-hmm.
I'll definitely be looking at, what are the common features between the two, and I'll probably look at HP1. That's a great idea. Thanks.
Okay. Good. Let's see. Do you see the RNA Pol II being loaded on the Kozak sequences instead of the promoter regions?
um, this is really embarrassing, but I don't remember what a Kozak sequence
That's all
is.
right. Rama, can you follow up and
I'm sorry.
clarify? That's fine. I don't either, so no worries. We'll
Is
come
that
back to that one.
okay?
Yeah.
Thanks.
Let's see. Did you check for increased microRNA levels?
Yeasts do not have microRNAs, but
Okay.
that is something that, but I will mention that there are other non-coding RNAs, like
Hmm.
cryptic unstable transcripts, and those actually are increased in the absence of risk. Um,
Okay.
so yeah.
What does that mean? So for like somebody like me, that's it.
Oh yeah.
Yeah. What does that tell you?
So it tells me that essentially, like, it's a lot of those antisense transcripts. They're like some short ones that
Hmm.
get transcribed that normally don't get translated. In fact, they're usually rapidly degraded. But I think there's a
Okay.
lot more getting created in this situation. And so again, like the transcriptome is super messed up. And so cells are not happy about that. But I can't look at microRNAs, but it's something I can do when I look at the mouse cells.
Okay. Let's see. Is the doubling time faster in Fun30 knockout?
Oh, that's a good question. I didn't look in log. Of course, they do exit faster, but I think I only looked at one cell cycle afterwards. So I haven't looked at that yet. So that's a good idea.
Okay. Did you use a pan-acetylated H4 antibody or specifically acetylated XH4 like K4 or K9 acetylated H4?
I used a penta-acetyl antibody. I haven't looked at the individual marks in this situation yet.
Hmm.
It's definitely something that would be interesting to look at for sure. And I'm probably going to go down that road at some point.
Okay. Okay, all right, good. Rama came through with the what a Kozak sequence is. She says it's the nucleotide motifs that are at the start of mRNA transcripts.
Oh, okay. Okay, so the question was where was polymerase? Thank you for reminding me of
Yeah,
that.
yeah. Do you see the RNA Pol II being loaded on those Kozak sequences instead of the promoter region?
Um, I didn't look very specifically at each transcript. I actually, that's a good analysis that I should have done. So the paper's already published, but something could be, so I could look and see, like, on average, how
Mhm.
far in advance is polymerase loading. That's a good something I could have done, and I could still do now.
Yeah,
um,
there's always plenty of things left to do. Sometimes,
yeah,
yeah, sometimes you just got to cut it off.
yeah. There's so many things messed up in the genome when risk is gone.
Oh yeah, I believe that. So
Mhm.
I guess so. That's all the questions we currently have. If anybody wants to get any in at the last minute, please feel free to do so. I do have one sort of. I guess it's a
Mhm.
little bit more general. Probably a little bit basic question. How
Mhm.
much? A lot of the work that you showed here, or maybe all the work here, was in a yeast, where it was with yeast cells. Um,
Right.
how well does that translate to other types, like human cells and that kind of thing?
Mhm. Yeah, that's a great question. And this is actually one of the reasons why I'm so interested in the chromatin questions.
Hmm.
And that's because the chromatin repression seems to be pretty well conserved. The Pattison lab actually just published a beautiful preprint looking at histone acetylation levels in quiescent glioblastoma cells. And they actually see that in the G0 cells in the tumors, those actually have a lot less H4 acetylation, for example.
Hmm.
And they even went far enough to figure out which histone lysine acetyltransferase mediated that. And so at least in GBM, it's conserved. And then there's another paper that looked at, I think, like B cell activation.
Hmm.
And those guys also have lower H4 acetylation. They also have risk bound to the chromatin there. So that's really cool.
Hmm.
Or the human version of risk.
Yeah.
So I think we're going to be seeing a lot of the similar, like, gene expression shutdown. But there's still a lot unknown, and I'm going to be looking more at, like, how are these conserved between mammals and yeast, and or just understanding each of them separately, because I think they both offer a lot of different ideas. Like quiescent yeast can be desiccated, for example. You can't really desiccate a human cell, I don't
Sure.
think. um,
Okay.
so there's a lot of interesting stuff there to look at, just basic biology wise, and then chromatin wise, I think there's a lot of very interesting conserved ideas that you can look at.
Okay. Very cool. So
Thanks.
I'll ask one last question if nobody else sends one in. What, whether it's 5 years from now or 50 years from now, what do you envision as the applications of this work? Like what will this turn into, I guess?
Yeah. So I'm hoping that someday you could use this to help with like cancer, for example. So if you have a quiescent cancer cell, it can evade a lot of therapies and maybe stick around and come back too. So I'm hoping that, looking at these fundamental mechanisms of regulating quiescence, maybe they can be applied to that.
Okay.
Especially looking in the chromatin area, there are already therapeutics being made for switch SNF complexes, for example. And so maybe we can see like, oh, will this be applicable to, tumors that have quiescent cell populations, for example. But also like the aging brain has quiescent neural stem cells, for example, and those guys have to activate and exit quiescence. And if that activation actually decreases with age. So I actually think there's a lot of aging, maybe anti-aging applications that can be used. So this quiescent idea is also a model for chronological aging. So that's something to look into as well.
Absolutely, that would be. I did not realize. That's very cool.
Apparently even like exercise can activate neural stem cells and stuff. So it's like an area I'm actually moving toward now. And so like the neural stem cell part. And so learning a lot of new things personally. So it's cool.
Well, that is super cool. That's really neat. Like I said, I had no idea. So that's really neat.
Yeah, I didn't either. Yeah.
That's awesome. Fantastic. So I think that's all the questions that we have. So if I get you to hang around for just 1 minute, we'll talk very briefly.
Absolutely. Yeah.
Thank you again to Dr. Cucinotta for a fantastic talk. This is super cool work. And thank you to everybody for coming out, asking great questions.
Thanks, everyone.
Be back again next week, same time, same day, or same day of the week, I guess. Another fantastic talk. Yeah, have a great weekend, everybody. Next week. Thanks for coming out.
Okay.